Hello everyone. Today we will be discussing the scientific paper titled Idiosyncratic Nutrient Requirements of Cats Appear to be Diet-Induced Evolutionary Adaptations. Let's take a look at why this is and how it can be explained using biochemistry. This paper is used to explore the unique nutrient requirements of domestic cats and to search for a reason as to why cats have these unique and specific dietary requirements. Given this, we will explore the evolutionary biology of domestic cats, the diet needs in relation to biochemical pathways, and the importance of a diet that closely mimics a cat's natural wild diet. Okay, moving on. Ooh, that is not supposed to be there. Before we get into the nitty-gritty, let's look at the timeline of evolution for domestic cats. Animals resembling cats appeared around 30 million years ago and looked something like this. However, modern cat ancestors didn't appear until the mid-Miocene period around 15 to 20 million years ago and looked like this. Around 15 million years ago, they began showing traits of true carnivores. Now, previously, these animals were obligate carnivores and ate only meat as a protein source. DNA studies of today's 37 cat species suggest they evolved from about 8 separate lineages in the last 10 to 15 million years. Most cat species we see today emerged relatively recently, with fossil records typically less than 2 million years old. The oldest fossils are around 3 to 5 million years old, and some are as recent as less than 100,000 years ago. Domestic cats evolved from the African wild cat, shown here, around 4,000 years ago, and here are domestic cats now if you needed another reminder. Let's take a look at a normal domestic cat's diet. The typical diet of cats is described as being high in protein, moderate in fat, and low in carbohydrates. This dietary profile reflects the natural diet of wild cats, which primarily consists of meat from prey animals. Like we said in the last slide, cats are obligate carnivores, meaning they require nutrients that are found primarily in animal tissues to meet their metabolic needs. Therefore, their diet needs to include high-quality animal protein sources to provide essential amino acids like arginine, as well as other nutrients such as arachidonic acid and certain vitamins that are crucial for their health. These, however, are not the only things that are crucial for a cat's health. Okay, but why the high protein? Can't cats just eat what they want and get rid of waste just like humans? Well, no. You see, unlike other mammals, cats have a limited control over the enzyme's activity in the urea cycle, specifically the activity of argininosuccinate lyase. This enzyme is responsible for detoxifying ammonia and converts argininosuccinate into arginine. Keep argininosuccinate lyase in mind because cats actually don't have this enzyme. As a result, cats need more protein in the form of arginine to efficiently dispose of this nitrogenous waste. The extra protein provides more substrate in the urea cycle for these enzymes to work with and keeps cats healthy. So, cats need high protein due to how their enzymes function, which affects their nitrogen metabolism and urea cycle. As you may remember from class, carbamoyl phosphate plays a critical role in the urea cycle. For your first quiz question, I want you to discuss how the activity of carbamoyl phosphate synthetase 1, CPS1, is regulated in the liver. I'll be looking for concepts regarding the general location of this activity, the general mechanism behind CPS1, and how CPS1 is connected with the malate aspartate shuttle. Remember, I'm just looking for these concepts in general, so unless you feel inclined, you're not required to go into depth into any of these. Section 18.2 of your book should help with this question, and try to answer this question before moving on. We're going to be discussing details of this question afterwards, and I think it's important to know what's going on before we talk about it, and you can add to your answer afterwards. Just try answering it beforehand. Alright, I trust that you all have at least attempted the first quiz question before moving on. Now, let's take a deeper look into the urea cycle. This is a figure from the book, and to review, the urea cycle facilitates the detoxification of ammonia, a byproduct of protein metabolism, by converting it to urea. The cycle is vital for maintaining a nitrogen balance in the body. Let's move through the urea cycle and then take a look at what makes cats different. To get to the urea cycle, we have to start in protein metabolism. When protein-rich foods are consumed, enzymes break down the protein chains into individual amino acids in your stomach and intestines. Some of these amino acids are used for building new proteins, enzymes, and hormones, but some aren't immediately needed. To handle these extra amino acids, the body goes through a process called deamination. Deamination is a biochemical process where the amino group, shown here in red, is removed from the amino acid molecule. This reaction typically occurs in the liver, where enzymes called deaminases catalyze the removal of the amino group. Once the amino group is removed, it's converted into ammonia, which is highly toxic to cells. That, my friends, is where the urea cycle comes into play. This picture shows where we left off. Ammonia. We are in the mitochondria about to make carboxyl phosphate, our first friend from the first quiz question. With the addition of carbon dioxide, ATP, and CPS1 to ammonia, we can, synthesize carboxyl phosphate. This, if you remember, is the irreversible step in the urea cycle, ensuring that toxic ammonia is removed. Once carboxyl phosphate is produced, the true urea cycle can begin. You can see in this picture that the amino group is highlighted in blue. As it's being passed through the urea cycle, notice this connection. Where does this molecule of aspartate come from? Well, I'm glad you asked. Do you remember our other friend from the first quiz question, the malate aspartate shuttle? Yeah, let's bring that back in. You cannot get away from the TCA cycle. Alright, you should recognize a good portion of this, like that the malate aspartate shuttle takes malate and pumps it into the mitochondria for the TCA cycle and pumps aspartate out. Aspartate enters the urea cycle behind argininosuccinate, making the malate aspartate shuttle key to the urea cycle, making our way through to arginine, which through arginase makes urea to be excreted. Earlier, we talked about cats' diet, so let's get back to that and tie this all together. Say it with me. Cats used to be what? Obligate carnivores. Today, domestic cats are still obligate carnivores, so they eat meat as a significant component of their diet for protein, but they may also consume small amounts of other food sources, such as plants or insects. When omnivores and herbivores, however, eat diets with different protein levels, the activity of urea cycle enzymes increases with higher protein levels and, conversely, decreases with lower protein levels, allowing them to conserve nitrogen for making non-essential amino acids. Makes sense, right? However, cats, given low protein diets, don't reduce urea cycle enzyme activity. Instead, they continuously convert ammonia into urea rather than using it to make non-essential amino acids. A benefit of this lack of regulation is that cats and other carnivores can quickly use amino acids for energy in making glucose. Do you see why I asked about the regulation of CPS1? It's all coming together. But what about that random aspartate molecule, you ask? Well, since cats are obligate carnivores, they cannot produce an essential amino acid, arginine, like humans can. Like I said in the beginning of this presentation, cats lack another key enzyme in the urea cycle called argininosuccinate lyase in the liver. Since cats lack that crucial enzyme, they can't convert this argininosuccinate into arginine, meaning they need to get arginine from their diet. This means cats need to consume a higher protein diet with all of the essential amino acids, including arginine, for the urea cycle to function correctly. Right, so cats have a unique diet since they lack a key enzyme in the urea cycle, but what about the other part of the title? Wasn't this paper supposed to be about that unique nutrient requirement being an evolutionary adaptation? Well, you are 100% correct, which leads me to our second quiz question. For the first part, why do you think this evolutionary adaptation came to be? For this first part, I just want you to give your opinion, and there is no wrong answer. For the second part, tell me how evolutionary changes like this happen. Think about DNA and RNA replication when answering the second part. If you need guidance, I found an article that might help, and I'll make sure to leave a link for you to use at your discretion. To conclude, cats evolved from cat-like animals over 30 million years ago, and their dietary needs have changed since. The absence of argininosuccate lyase in a cat's urea cycle influenced their unique nutrition requirements. Since cats can't produce arginine on their own, they need a high-protein diet to receive arginine from their food. This is actually an evolutionary advantage that is shared with other carnivorous animals. Thank you all for bearing with me, and I hope you have a perfect rest of your day.